to visit with us virtually and give a talk on computational biology. Professor Jiang really started his bioinformatics or computational biology career earlier than most of the faculties who are active today, and he's still very active in computational biology research. If you look at his CV, you'll be very impressed with the foundational work that he had made in computational biology. So he got his bachelor degree in 1984 from the University of Science and Technology of China, and got his PhD degree in computer science from University of Minnesota, the same school that I also graduated. So I'm happy to be associated with him. In fact, we've known each other for over 15 years or 20 years. He was a faculty member at the McMaster University starting in 1989. That's over 30 years. And now he's a university distinguished professor at the Computer Science and Engineering at the University of California, Riverside. He has been very instrumental in founding some of the prestigious conferences in bioinformatics like RECON and ISMB. He's a fellow of the Association for Computer Machinery and a fellow of the American Association for the Advancement of Science for his contribution in combinatorial algorithm, computational molecular biology, bioinformatics, and computational aspects of information retrieval. And he's well recognized as one of the earlier contributors of our field. And I'm pretty sure that with us being in the medical school, and sometimes we focus too much on the applied aspect of bioinformatics, it would really be eye-opening for us to hear a different aspect of computational biology and bioinformatics. So without further ado, let's welcome Professor Tao Jiang.
Okay, well, thanks so much to Jake and Amy for the introduction and for the invitation. I had wished that this can be made in person, so I actually get to see your beautiful campus. But since the pandemic doesn't seem to be ending anytime soon, I guess a virtual presentation is the next best thing. So we're coming from slightly different background, and also, given this this remote presentation, I hope I can make the make this as informative and also enjoyable as possible. So today I'm going to share with you some of our recent work on the prediction of isoform functions by combining powerful techniques from machine learning and also gene interaction networks. This is a joint work with some of my former students, Hao Chen and Dipen Xiao, who are now postdoc and the researchers at CMU and Illumina. And also this was collaborated with Jiang at Tsinghua University, Dongbo Bu at the Chinese Academy of Sciences. I don't know if I can see your chat, but if you have any questions, feel free to speak up, or maybe you chat and Jay or Amy can probably stop me to answer questions. Let me start by repeating this central dogma from molecular biology, even though I'm pretty sure this audience doesn't need it. Hopefully it's not too insulting. So we know genes sequence of exons and introns, and the transcription process copies both into a primary RNA or pre-mRNA, and then the splicing process removes some introns to produce an mRNA. What's interesting is this splicing process is not a unique process. For example, some introns can be retained and some exons can be skipped. Because of this, we have this phenomenon of so-called alternative splicing, and the products of alternative splicing are called isoforms. So as you can see here, in this example, we have two different isoforms by choosing different exons from the primary transcript. So we have isoforms at the RNA level. They are simply different mRNA transcripts. We also have isoforms at the protein level. There are different protein sequences with probably different structures and potentially different functions. The central question we're interested in today is what are the specific functions of these isoforms? Unfortunately, very little is known about this question, even though there has been some research evidence that the isoforms of many different genes do have different functions, and this difference in functions actually is important for biological processes such as disease. Let me give you a few examples. So this is in cardiovascular disease. The gene titin has 3 different isoforms. The 2 shorter ones shown on the left are stiffer, are expressed in normal hearts. The longer one shown on the right is more distensible, and its expression often leads to heart disease, the so-called dilated cardiomyopathy. The impact of misregulated alternative splicing patterns has also been found in neurological disease. For example, in Alzheimer's disease, many genes have been found to have specific isoform expression patterns that are directly linked to the disease. And this link between splicing and cancer has also been observed in splicing and disease has also been observed in cancers. So as shown in this pie chart, many genes have very specific splicing patterns that are implicated in important biological processes and properties, as shown with the color background, that play roles in cancers. And because of this, some isoforms have been used as biomarkers and also as therapeutic targets in cancers. So it's known alternative splicing is very common in many organisms, including human. 95% of human multi-exon genes undergo alternative splicing. Only 5% of them don't. And this explains the huge difference between the number of human genes, which is around 20,000, and the huge number of isoforms in human, which is already known to be more than 80,000. Gene functions have been studied by researchers for many years. So there are established experimental protocols and computational methods for determining gene functions. For example, we can modify a normal gene by using experiments such as knockout or knockdown and observe changes in phenotype. From these changes in phenotype, we can infer critical clues about the function of the gene. We can also analyze the evolutionary history of genes and identify the orthology relationship between genes. It is commonly believed that orthologs in different species often carry the same or similar functions. So what this means is we probably can infer the function of a gene in one species from the functions of its orthologs in other species. This knowledge of gene functions has been cataloged in several databases, and probably the most famous one is gene ontology, or GO. In GO, the functions are represented as so-called GO terms or GO terms. And the GO terms are divided into 3 main categories, also called branches, as shown in the 3rd column of the table. So the 3 branches are molecular function, biological process, and a cellular component, right? So these are some examples of GO terms and the corresponding biological functions. For convenience, the Go terms are organized as a directed acyclic graph, or called DAG, as shown on the right. So in such a DAG, the higher terms are generally more represent more general functions, and lower terms represent more specific functions. So what this means is, if a gene is annotated with a lower term in the graph, they should also have functions represented by higher ancestral terms. Okay, here's an example. So in this case, this gene is known to have this term, right? Basically, by inheritance, should also have all these higher terms because they are ancestors of this lowest term. As mentioned before, we know the isoforms of the same gene, of some genes, carry different functions. In fact, in some cases, the isoforms of the same gene carry very different, completely different, or even opposite functions. And here's an example. So this gene, CDKN2A, is very important in cancers. It has two different isoforms, and they have different structures as shown here. And we know they have completely different functions as represented by different sets of geo terms as shown in the table. But the reality we're facing here is, for most of the isoforms that we're interested in, we don't know their functions. We don't know their geo terms. Okay, and this situation is pretty bad because we know some of the isoforms actually play roles in important biological process such as disease. And here's an example. This gene ACMSD is very important in neurological disease. It has two different isoforms, and we know these two different isoforms actually play different roles in different neurological disease. However, up until now, we still don't know the specific functions of these two isoforms. Okay, so a natural, interesting computational question is, can we develop some algorithms to infer functions of isoforms? That is the question we're interested in today. A very simple formulation of the problem is, suppose we're given some biological features of isoforms. We're also given the functions of genes as geo terms. Okay, can we predict functions of isoforms as geo terms? Here, by biological features of isoforms, I mean things like sequences or expression profiles. Okay, more specifically, suppose we're given a gene that has a number of isoforms, and suppose we know the gene has been annotated with a bunch of Go terms. Okay, do we know for each Go term which of these isoforms actually have it? For example, maybe the first two isoforms have the first Go term, the first and third isoforms have the second Go term, and maybe all isoforms actually have the function represented by the third Go term, right? So this kind of assignment is something we like to know. Right. As a person who works on algorithms, we always like to start with a problem definition, and this is our formal definition of the problem. Does anyone have any question? In order to understand algorithms we discuss, we have to understand what the problem we're facing exactly. So if anyone has any question, please let me know. All right, so so this is the problem we're facing today. Now, if we focus on a particular Go term, right, then determining which of the isoforms of the gene actually have this Go term is a very simple binary classification problem. Even though it looks simple, but there's a big challenge. That is, we mentioned before, we don't have the ground truth information about most of the isoforms. So there's a lack of isoform level ground truth. So what this means is, the traditional supervised learning methods for gene function prediction or protein function prediction are not applicable. Just to show you how much is lacking in the ground truth, let me compare the functional annotation of genes that we have with the functional annotation of isoforms that we have today. Okay, so this big pie shows we have almost 500,000 geo annotations for genes. Okay, and the black means experimental evidence, and blue means manual review, and the gray means electronic evidence. So, for example, inference based on evolutionary studies. The quality of the red and the blue evidence obviously is better. It's more convincing, right? You can see that for gene functions, most of the annotations are based on experiments or manual review. However, for isoforms, most of the functional annotations are based on computational inference. Okay, well, even worse, even though we have far more isoforms than genes, but the number of annotations we have for genes, is much more than the annotations for isoforms. Okay, since about 10 years ago, some computational methods have been developed for inferring isoform functions. Here I listed most of them. These methods all use gene expression, isoform expression profiles, okay, based on the assumption that isoforms that have similar expression patterns should have similar functions. Right. So in these methods, we start from RNA-seq data and the gene ontology database. Then from these sources, we extract expression profiles for isoforms. Then we extract the ground truth labels for genes. Then by using a semi-supervised learning approach, in particular, this multiple instance learning framework, we infer isoforms. Okay, so multiple instance learning, or MIL, would allow us to deal with the lack of ground truth situation. However, there are many other pieces of information that can be very informative for isoform functions, but they are not used here. So our first idea is to incorporate these potentially useful pieces of information into the picture. For example, we also have the, for each isoform, we also have the amino acid sequences, right? And we know such a sequence typically contains many functional sites and signals, as shown here. And these functional sites or signals usually provide functional information directly, right? And also these sequences contain conserved domains. So these are domains conserved across different species, and these domains are often functional. They may also provide critical information about the functions, right? So a natural idea is, why don't we incorporate amino acid sequences or protein sequences of isoforms and also conserved domains into the inference algorithm, right? A difficulty here is these pieces of information are very different from expression profiles, right? So the first challenge is, how do we integrate these heterogeneous information? So the first work I'm presenting here is called DIFFUSE. In this algorithm, we use a two-stage process to integrate heterogeneous information, heterogeneous data. First, we use a deep neural network to capture features from amino acid sequences and conserved domains. As you can see in this diagram, we use the CNN to extract features from protein sequences. We use LSTM, that stands for long short-term memory architecture, to extract features from the domains. The domain, then the features would be concatenated and fed into a dense neural network. And the dense neural network will give us an initial score that indicates the likelihood for the isoform to have some function. This is the first stage. Okay. Then in the second stage, we try to incorporate expression profile information. So the expression profiles will give us the co-expression relationship. Okay. And I see there's a chat. Yes, so the question is there a bigram or a trigram used here? So actually, yeah, so the protein sequence would be represented as a set of trigrams. Yeah, skip that. All right. So so here we use conditional random field, or CRF, to incorporate the expression profiles. Okay. In such a CRF, the vertices are isoforms and the edges represent co-expression relationship. Then by optimizing a suitably defined Gibbs energy function, we will encourage isoforms that have co-expression patterns to have similar functions. Right. So the initial scores from the first stage will be fed into the CRF, and then we, by optimizing this energy function, we'll refine the scores to produce the final score. Okay, and as we mentioned before, we don't really have the ground truth of isoforms, right? So again, we use a semi-supervised algorithm, multiple instance learning, to help training. And here's an illustration of some of the details. So in this MIL framework, we think of genes as bags, and then we think of isoforms of a gene as instances of the bag. So a positive bag would be a gene that has a function, and a negative bag would be a gene that does not have the function. Okay, so we initialize the labels of isoforms, right? The instances the same as bags, as you can see here, right? So all the isoforms of a positive gene would be assigned a positive, and vice versa. All the isoforms of a negative gene would be assigned a negative, right? But during the training process, we allowed isoforms of a gene to have a different label as the gene label. Okay, as you can see here. So eventually we try to infer the functions of every specific isoform. So there's a question whether we use the embedding algorithm. Not in this work. In the second algorithm, actually, we used embedding to extract features. But these are some of the good, these are good questions. All right. So this is the sort of outline of our first method, diffuse. We have implemented this method and tested it on three different data sets. The first are human data sets with different number of isoforms and expression profiles, and the third one is a mouse data set. Okay, and in this evaluation, since we don't have the isoform level ground truth, we perform evaluation on the gene level. So we aggregate basically predictions for isoforms, right, to the gene level, and we evaluate on the gene level. As you can see from the table, our method, right, our new method, diffuse, performs much better than the previous method in terms of both AUC and AUPRC. In terms of AUC, I think the improvement is at least 15%, and in terms of AUPRC, the improvement is at least 80%. Right. So what this means is adding this additional pieces of information, right, amino acid sequences and the conserved domains really help prediction a lot. So we actually performed this the so-called ablation study to assess the contributions of the individual components. So what we did was to remove expression profiles or conserved domains and sequence features from the method one by one, and observe how much the performance would be affected. From the diagram, you can see that every component actually contributes to the performance of the method. So every feature used here is critical. Okay. In particular, the sequence features probably contribute the most, because if you remove sequence features, then the performance of the method would dip the most. This is interesting because the conserved domains actually were extracted from sequences, and they were supposed to have the most functional information, right? However, from this analysis, you can see that the conserved domains cannot completely replace sequence features. Yeah. So in the previous figure, these are all averages of the measures. They're simple averages of all the numbers. All right. So so we know all these features contribute critically to the performance of the method. We also analyzed the contributions of individual size or individual positions of a protein sequence to the performance. So we use a technique called saliency maps to assign the so-called importance scores to individual amino acids. Right. So the importance score basically would represent the weight that a particular position has in the neural network. Then we take average of these importance scores inside conserved domains and outside conserved domains. As shown in this diagram, you can see that on average, the conserved domains contribute more than regions outside conserved domains. Right. So if you compare this with the previous slide, the conclusion is the conserved domains do not completely replace amino acid sequences. However, they still contribute more compared with the non-conserved domain areas. Right. We started by assuming that isoforms of the same gene do have different functions. So here we like to confirm this. Right, so analyze the divergence of the predicted functions of isoforms of the same gene. We use a tool, GOSSTO, to assess the semantic dissimilarity between functions assigned to different isoforms of the same gene. Okay, and we look at the three main geo branches separately. As you can see in the figure, in every branch we find many genes to have isoforms with very different functions. Right. So this is a confirmation that isoforms of the same gene do have very different functions, at least according to our prediction. Okay. The biggest challenge in this line of research is to validate our prediction results, right? Simply because of the lack of the ground truth at the isoform level. So we did the next best thing, that is we did some indirect validations. Okay, first we looked at the correlations between functional sequence expression similarities. This is more like a sanity check than a validation, because we expect isoforms that have similar sequences or expression profiles to have similar functions. We like to double check whether that's true in our prediction. Okay. As you can see in the diagrams, we do find a moderate correlation between functional similarity and sequence similarity, and also between functional similarity and expression similarity. Okay. Interestingly, the correlation between a function and a sequence is higher than between function and expression. And this is consistent with our previous observation that the sequences would contribute the most to the functional prediction. Okay. Further, since we know protein structures are more conserved, we would expect isoforms with similar functions to have more similar structures than sequences. Okay, so we like to calculate the correlation between functions and structures and see whether we find a stronger correlation than the previous correlations. The difficulty here is, how do we get accurate structures of isoforms? And remember, this was done before powerful tools such as AlphaFold was available, right? It's very, very computational intensive to predict three-dimensional structures of isoforms, even if we trust the computations. So we used contact maps to approximate protein structures. Okay, and we use the overlap between contact maps as the structural similarity. So we calculated the the correlation between functional similarity and structural similarity as contact map overlap, and compare that with the functional similarity with sequence similarity. Right, the numbers have changed a little bit because this is a smaller data set. We're not able to get contact maps for all the isoforms, tens of thousands of isoforms. I think this data set is slightly over 1,000, about 1,500 isoforms. And you can see that the correlation between function and structure is significantly higher than the correlation between function and sequence. And this is consistent with with our hypothesis. So the first question is, do we have any tissue-specific information? So in this work, we look at all the tissues. We look at all the general data that combine many tissues. But in the second part of the work, I'm going to talk about some tissue-specific information. Okay, t-SNE of PCA, clustering of embedding of figure. No, the short answer is I don't believe we did. Okay, but I'm not clear exactly what you're getting to. So clustering of your embedding or figure out any pattern. I guess you're trying to ask whether we see any patterns of the features.
Yes, sir.
So as I mentioned, for diffuse, we didn't do any embedding. We could have done that probably by looking at the layers of the let me show the network here. Yeah, so we didn't do any explicit embedding, but potentially we could look at the layers in these neural networks and maybe do some analysis as you suggested, but we didn't.
Yeah, in that way we can, like, in my opinion, we can get important feature and understand how this model is working actually. Like what components are important to make the prediction, right?
Well, that was done actually by using the ablation study, right? So we peel off these components and see how the performance would be affected.
Well,
yeah, yeah. So, but we also use Salinsy maps to assign important scores. So that will give us an idea what positions of the sequences actually contribute the most, right? But we don't look inside of the model to find more patterns, that if that's what you're suggesting. Oh, well, thank you, Steve. I guess we can probably do either. I mean, whatever you prefer. Right. So, all right. So let me proceed. I guess maybe I need to speed up a little bit. All right. Okay. So I think this is the final validation. We mentioned the sequences, the amino acid sequence may contain functional sites or signals, and these signals or sites actually give us clues about the functions directly. It turns out this famous database UniProt contains some sequence annotations, sequence feature annotations for some of the isoforms we analyzed, right? For example, if we know isoform, and we can use such sequence features to perform some indirect validation. For example, if we know isoform 1 has a particular sequence feature and isoform 2 has another sequence feature, then we can also predict functions according to these sequence features. All right. So the number of isoforms that have annotated sequence features is not so many. We collected all of them. Okay, and then we compared the sequence features, their sequence features, with the predicted functions by these different methods. In this table, the circle means a positive instance, and the cross means a negative instance. Okay, and we use Jaccard index to measure agreement between sequence features and predictions. You can see that our method, DIFFUSE, has the highest agreement. Right, but this is a very small set of validation with a small number of features as well. All right, so let me move on to our second second algorithm, where we try to incorporate gene interaction information into the prediction. Okay, so we know proteins usually do not work alone. They interact with other proteins to perform function. And the interactions of proteins form a network, the so-called protein-protein interaction network, or PPI. Okay. Interestingly, the proteins that perform the same function often form subnetworks in the network. Okay, so the idea is maybe by using the subnetwork structure, in particular the neighborhood information of PPI, we might be able to help predicting isoform functions. Right, so this is the question: how do we incorporate interaction information to enhance prediction? Well, the difficulty here is the current PPI network information is all built on gene level. On the other hand, there has been some previous research that shows that the isoforms of the same gene may often interact differently. Right. So according to this paper in Cell some years ago, most of the genes, I guess in human, have isoforms that interact quite differently at the intermediate level. Right. The dilemma is the PPI information we're given is all at the gene level. Right. So how do we resolve this issue? So our solution is, if we cannot solve one problem, we solve two problems. Okay, we propose a new method called Refiner, where we try to predict isoform functions and refine PPIs at the same time. So here by refinement, I mean we try to refine the PPI network at the gene level to isoform level. At the end, we obtain the so-called isoform-isoform interaction network, or III. Okay. This is our model architecture. There are three modules. The first module is similar to DIFFUSE, where we try to combine sequence features with expression features to predict isoform functions. Okay, but the difference is actually we do some explicit embedding. Okay. And instead of using LSTM, we use RNA, a more general recurrent neural network. All right, and in module C, we try to refine the PPI into III. Right, so we assume we're given initial network at the gene level, and at the end, we hope to have the interaction network at isoform level. Then in this middle module, we have a mutual regularization that will encourage isoforms that interact similarly in the network to have similar functions, and vice versa. Isoforms that have similar functions will interact similarly in the network. Okay, so this is the basic idea, and to test the performance, we use some tissue-specific data, as mentioned, as asked previously. So there are totally 15 data sets. We have 12 major issue data sets and also 3 brain tissue data sets. We also compared this method with some previous methods for predicting tissue-specific functions on net, as well as the best isoform function prediction method, including DIFFUSE, we just mentioned before, and there's a newer method called DISOFUN. Okay, in order to assess how the refinement of a network contributes to the performance, we also looked at some restricted versions of Finer, where Finer fixed raw means we simply use the initial given a PPI network at the gene level. Okay, we do not refine it. And in Finer fixed NE, we use a fixed network, but the network has been refined from the initial network by using the best network refinement method called a network enhancement. Right. Finally, in Finer, we dynamically adjust the network according to the functional prediction. Okay. As you can see in the table, by using network information, we're able to improve performance somewhat, right? Both in terms of EUC or PRC. But by allowing the network to dynamically adjust, we are able to improve the performance a lot. Right? I think in terms of EUC, the improvement is about 8%, and for EUPRC, it's a bit more than 8%. It's more than 10%. Okay. To convince you that this joint learning of both functions and networks really makes sense, we look at the tissues. We look at the data sets individually. Right, so in the histogram, every bar represents basically a different data set. You can see that, and orange means the full model, right? This is with the network refinement, and the green bar. I think we can skip the blue bar, and the green bar means without the network refinement, right? You can see that for some data sets, such as this one and this one, the refinement makes a huge difference. Okay, and also as shown in diagrams on the right, by doing both simultaneously, we're able to cluster isoforms that have similar functions as a subnetwork in the network, and this makes more sense. All right. So then we also take advantage of the available tissue information of geoterms, and we try to validate the consistency between the predicted isoform functions and tissue specificity. Okay, so what we did was we divide isoforms into tissue enhanced and non-tissue enhanced isoforms based on expression level. Okay. Then we compared fold enrichments of tissue-specific geoterms in both tissue-enhanced and non-tissue enhanced sets of isoforms. Okay, as you can see in the table, in most cases, I think in almost all cases except for one or maybe two, right? We have more enrichment of tissue-specific geoterms in the tissue-enhanced isoforms. So in other words, there's a consistency between the predicted functions and the known tissue specificity of the geoterms. Okay. Then we also checked how isoforms interact with each other with respect to their subcellular locations. Okay. It turns out there's some previous study in the literature that provides subcellular locations of the isoforms we studied in our data set, or some of the isoforms we studied in our data sets. Okay, and intuitively, if isoforms are located in the same location, they are more likely to interact with each other, right? And so basically, as you can see here, if we divide the location into 3 categories, then we would expect interactions to be in this diagonal form, right? So here 6 means single isoform genes, right? Remember, we're given the network information at the gene level, and if there's only one isoform in the gene, then that network is also the isoform network, right? So if the given network is high quality, we would expect the network to look like this, right? However, for multiple isoform genes, the given network actually is at the gene level, so probably has a lot of noise with regard to isoforms. So maybe the initial network is not perfectly diagonal, but after refinement, we would expect the network to have this diagonal pattern. So this is our results. All right, and this is for all the tissues. You can see that for single isoform genes, the initial given networks were almost perfect, right? But after refinement, they're even better. Okay. However, for multiple isoform genes, the initial networks are very noisy. Okay, they're probably not noisy for genes, but they're pretty noisy with respect to isoforms. But after refinement, they're much better, right? They follow diagonal pattern much better. Right, so this is a strong validation of the quality of the refined networks with respect to subcellular locations. Okay, so I think this is the final page. So we also try to validate the functions, predicted functions, with respect to subcellular localization information. So what we did was we first identify a set of GOI terms that are specific to certain locations, as shown here. Okay, and we only consider multiple isoform genes or mix that have isoforms with different localization. right? Then for each such tissue-specific geo term, we quantify the agreement between isoforms with predicted functions and isoform with the at the corresponding subcellular locations. And agreement is measured by using a Jacquard index, right? So as you can see in this table, our method, FINER, right, was able to have the most agreement in most cases, in almost all cases except for one where DIFFUSE actually did better, right? All right, I think my time is up. The work I described today has been summarized in 3 publications. They appeared in journals Bioinformatics and AR Genomics Bioinformatics, right? In particular, the tools I mentioned, Deep isoform, DIFFUSE, and FINER are all available on GitHub. So if you're interested, feel free to download and use them. And the data sets I mentioned are also available there. As for future work, I think next we will try to incorporate protein structure information into the prediction. It was impossible before because it was very difficult to obtain high quality of structure information for all isoforms, right? But now, given the availability of AlphaFold or Rosetta Fold, has become reality. And I think the structural data will help improve the performance significantly. The second is we should try to take advantage of the growing volume of single-cell RNA-seq data, right? These data typically come with spatial temporal information, and as we have shown before, such information, such as subcellular localization information, can be used for validation, right? In all these algorithms, we represent functions as geoterms. But there are other forms of functions. For example, the participation of a gene in the pathway, right, and interaction with non-coding RNAs, right? These are all functions of genes. Right? Interesting question is, can we also study isoform functions in terms of these functions, right? What's interesting is, we're also given such functions. There's a lot of database that provide such functions at the gene level, right? So our main task would be again to refine such function information at the to the isoform level, right? We'd like to know whether the same frameworks we discussed today can be also applied, or they have to be modified. I guess that's all I have to say today. Well, thank you very much for your attention. If you have any questions, I'll be happy to answer them. Thank you.
Well, this is really amazing. I think every time I talk to you and you come up with some really fundamental work that that is going to be profoundly used in the field. I think there's a question from Zoom now. Yeah, if you can.
Right, so I see a question on chat. Yeah, so we in this work we measure functional similarity using semantic similarity or semantic dissimilarity.
Okay, great. So so
how trustworthy is semantic dissimilarity? Well, here we trust the gene annotation, right? Remember, the gene annotation is done using Go terms, and this is something we trust. I guess your question is if they are annotated with different Go terms, right? Does semantic similarity really represent functional similarity? That's something we don't know. So here we have to trust the geo-annotation of the gene functions.
And next is whether there's a specific cutoff that can apply to say this similarity is accept, and then some of the similarity.
Yeah, so that we don't know. So we simply plotted different dissimilarities. So as if I can show you, we divide the whole range into 20 bins, and we simply plotted situation. Right. So in this case, we divide the range into 20 bins, and we show the number of genes each bin. So as for cutoff, I guess probably can try different cutoffs depending on application.
I see. Thank you.
Yeah, so I want to expand on the previous question and ask what's your thoughts on this? So essentially, right now you're using the gene ontology annotation, which is actually probably going to take some time for people to make note of that in the database. A lot of the time when we're doing the biomedical research in the medical school setting, we're into the new territory. So for example, if we look at the cancer treatment before cancer patient expression profile before treatment and after radiation treatment, we see new alternative splicing patterns. And so there's a lot of metadata in the experiment itself. So can you extend your future algorithm to allow taking the metadata of the experiment before and after and the impact on the isoform to be used as a label in your algorithm?
Yeah, that's a very interesting question, and this is related to sort of the third part of the future work I mentioned. So yes, it is true. All these algorithms are based on gene ontology, but all we require here is a functional annotation of genes, right? So if from your experiments you are able to obtain some functions for genes, then we can still, apply the same algorithm to refine them into isoforms. That can be done, right? And if your experimental data even suggests something more specific about isoforms, that information can be potentially incorporated. But I guess all we require is some kind of gene level annotation of some function.
Yeah, I have another question before we go to Tom. So my other question is, in your current way of doing the deep learning, do you assume a somewhat independent effect of the different parts of the isoform? What about potentially synergistic effect? For example, inclusion or deletion of a module would actually cancel out the functions of another module. Did you actually see that that kind of effect is able to be captured in your prediction and is accurate?
That's also a very interesting question. The synergy, right? The cooperation of isoforms. I think this may have more to do with the second work where we use the network information. So right now we only use a network, PPI network, as a hint of functional similarity, right? So if they have a lot of links together, then they probably have similar functions. But we have not considered the cooperation of genes in performing functions. That's something probably. So probably you're talking about finding functions of modules functionally in a functional modules, right? So that's probably another thing we can do with networks. Yeah, but we have not considered that in our work.
Thank you. So there's a last question, go to Tom Broker. He typed in the chat box.
Have you included human virus genomes in your analysis? The answer is no, we have not. Viral proteins and their expression patterns often serve as the best documented structure-function relation, as well as viral proteins.
Yeah, so I wanted to elaborate on that because I really strongly recommend using them as a tool for understanding not only the various structure-function relations, but the alternative splicing within viral proteins is extraordinarily elaborate. and partitions functional domains regularly into different portions of transcripts, which can be incorporated into multiple other proteins as a result of the splicing. The second thing I wanted to emphasize is that a number of genes, including those in viruses which we study, use alternative promoters and alternative polyase sites, as well as the internal alternative splicing. So there's additional levels of complexity there.
So by the viral proteins, you mean the proteins that interact with the virus?
Well, both ways, but a lot of the way, for example, adenoviruses or papillomaviruses interact with host cells to modify their functions to support the viral persistence and replication, for example, involve just extraordinarily elaborate expression patterns. But because these same viruses are pathogens, they've been heavily studied so that one can understand a lot of structure-function interactions related to cancer, to cell cycle regulation, protein assembly, and so forth. They're an incredible wealth of information, and the value is that they have relatively small genomes ranging from 5,000 base pairs to, say, 100,000 base pairs. So they compact a lot of information into a short genome, and as a result, they have to depend on a lot of alternative processing, synthesis and processing, to achieve all the same interactions that a lot of the cell itself will require 20,000 genes to do.
Okay. Well, this is very interesting. I didn't know that the virus proteins actually genes go through splicing, alternative splicing process, because they're.
If I can elaborate on that, my wife Louise Chow and I are the people who discovered splicing and alternative splicing, and we reported on it extensively in 1977.
Oh, okay.
So there's a vast amount of work that went on in viruses for 20 years before people even understood what was going on in the eukaryotic genes.
Right. Okay. Well, it's good to know. I will try to look the information up and try to understand more what's going on there.
Well,
thanks for the suggestion.
Yeah, contact me, because we have 50 papers on alternative splicing and the assignment of functions to the different portions of the mRNA and the proteins.
Oh, okay. For sure, I'll send you an email.
Okay.
Thank you.
Okay. Well, I think Professor Jiang is right, and he really have wished that he could come in person. Then we'll have more time to talk about interesting topic like this over lunch or in the afternoon. But it is what it is, and I think one thing I can tell you, Professor Jiang, is your talk has attracted the most number of questions that I've seen in years. So it shows that you have done a fantastic job in simplifying the very advanced algorithm work to make it easier to understand by people in the medical school, and we have lots of potential application domains. I took the liberty of sharing your email here in the chat box. Those of you who are interested in collaborating, perhaps they can send email to you, if that's okay.
Sure, yeah. I will also try to look up Tom Broker's, maybe you can also send me Tom Broker's email. I'll definitely try to get connected with him.
Okay. All right. Well, thank you so much again. And this is a wonderful morning. Yeah. And,
okay, well, thanks so much to you guys.
Okay.
And I hope to see some of you in person sometime in the future, in the near future, eventually.
We'll see you at ISMB in Madison, Wisconsin.
All right.
Okay.
Okay.
All right.
Bye-bye. Have a good day.
Bye.